Okay, welcome back. Let's start taking a look at microtubules. And like I said, this section will be in a number of parts, just to break things up and make the video shorter. So microtubules are the largest structural elements of the cytoskeleton. They've got the greatest diameter. They're involved in a number of functions in the cell, which we'll start looking at in a moment. They pervade the cytosol and are responsible for things like maintenance of axon structure in neurons. They're involved in the formation of meiotic and mitotic spindle. They maintain and alter cell shape, and they can be responsible for moving and placing vesicles using motor proteins. So that's cytoplasmic microtubules. Axonemal microtubules, those are more organized and stable, and you commonly find those in structures like cilia and flagella, or a structure called the basal body, which is often found at the base of an axoneme, such as a cilia or a flagella. And so those are much more organized, and they often work with many accessory proteins, including motor proteins. So if we take a look at the basic building block of the microtubule, we find that there are two types of tubulin protein that make up the microtubules: alpha tubulin and beta tubulin. And the alpha and beta tubulin have nearly identical three-dimensional structures, like you can see here when you look at the structure of alpha tubulin and beta tubulin, and hopefully you recognize alpha helix and the beta sheets in the structures of alpha and beta tubulin. So virtually identical structures, yet they only have 40% amino acid identity, and so that is because of the conserved nature of the amino acids that you find in these proteins. And so even though you've got two different amino acids, R group functionality can be conserved because of the chemical characteristics of the R groups that you find in different but functionally conserved amino acids. Each of the two tubulin isoforms has an N-terminal GTP binding domain, and then a central domain to which a drug called colchicine can bind, and then a C-terminal domain which is involved in the binding of proteins called MAPs, or microtubule associated proteins, that can modulate the activity of microtubules. When you look at a protofilament, that's one of the long filaments that makes up the microtubule, all of the dimers of alpha and beta tubulin, these dimers down here, they're all oriented in the same way, with an alpha tubulin towards the minus end and a beta tubulin towards the plus end. So down here we've got the actual straight hollow cylinder. The length varies considerably in the cell, and this microtubule contains approximately, usually, 13 of these protofilaments, which is a long chain of alpha and beta tubulin dimers. These dimers are associated non-covalently, so the dimer is really the functional subunit which is used to build the protofilament, and the protofilaments assemble to form the hollow microtubule. As you notice, there is an N negative end and there's a positive end, and so this microtubule has some polarity, and we'll look at the polarity in the cell right now. So if we look at the organization of microtubules in the cell, we can look in interphase and we can look in mitosis. Here's Eddie coming to say hello. He likes microtubules. So if we look at that top in part A, that top figure, top left, part A, we can see that that's a cell in interphase. Excuse me. And we're looking at the distribution of microtubules there using confocal fluorescence microscopy. So in the top we've got a cell in interphase, and at the bottom we've got a cell in mitosis with a mitotic spindle. So the microtubules, they always radiate from a place called the microtubule organizing center in the cell, and in animal cells that's most commonly a pair of centrioles that form a centrosome. Centrosomes are absent in plants, but there's a region, there's a region of cytoplasm that acts as the microtubule organizing center, and so that region of cytoplasm in plants has the same function as the centrosome in animal cells. So in the interphase cell, you can see that the microtubules are radiating from this point here, close to the nucleus, which is kind of a little bit shadowed here, and they then radiate out through the cytoplasm. So they radiate from one point in an interphase cell. During S phase of the cell cycle, the centrosome duplicates, and we have two microtubule organizing centers, one at each pole, and then these become the spindle poles. And sometimes these are called the spindle pole bodies in dividing cells, in mitotic cells. So then all the microtubules radiate from the centrosomes or spindle pole bodies. There are two populations of microtubules here, probably three populations really. There's the mitotic spindle population, which is the population in the middle. Then these ones towards the end, those are astral microtubules. And we'll talk about these more when we talk about cell divisions. Now, in the diagrams here, what I want you to pay attention to is the polarity of the microtubules. Here's a nucleus. It's a little bit small compared to the scale, given the size of this cell. The centrosome, this pair of centrioles, is right here, close to the nucleus. The minus end of the microtubules is embedded in some pericentriolar material, which is just material that's around the centrioles at the centrosome. The negative end is embedded there, and the positive end is radiating out towards the edge, the end of the cell. In the mitotic cell here, the centrosomes are the spindle pole bodies or the spindle poles, and the negative ends of the microtubules are anchored there, and the astral microtubules, the positive ends are towards the cell's plasma membrane, and the microtubules that are forming the spindle, the plus ends are towards the center, towards the metaphase plate of the cell. So that's the organization, and make sure you take note of the polarities of the microtubules. Microtubules can be singlet microtubules, so that's just a hollow filament, 13 protofilaments making up a singlet. They can be these kind of fused structures where you have one complete ring and then another partial ring, so that would be a doublet of microtubules, and those are commonly seen in cilia and flagella. Or you could have a triplet where you've got one complete ring and then two partial rings, and we often see those in basal bodies and centrioles. So one full ring and then usually 10 to 11 protofilaments in the partial rings. So that's it for the first part of microtubules. I'll be back for the second part. I'll see you soon.